Welcome everyone to week number 3 in Biology 120 lab. We've got a lot going on this week, so let me get right into it as soon as I can. There we go. Okay, so last week we covered pH, which is a measure of how acidic or basic a solution is, and buffers, which are chemical systems that maintain pH. And we learned about why pH is important because most organisms love a nice neutral pH. You change their pH, you change the pH of their environment, and they don't like it. That's why ocean acidification is tremendously important as a part of climate change, because we are changing the pH of the ocean, and that means we will harm organisms that live in the ocean. Buffering systems are terribly important in organisms because we have these chemical systems set up that keep our pH nice and neutral, because that's where our cells want to be. And when those get off, then we have a lot of trouble. In fact, we can die from that if our pH gets off balance. All right, this week, what are we going to be learning about? We're going to be learning about diffusion and osmosis and cells. So, we got a lot to cover. Diffusion and osmosis both have to do with cells and how cells work. So we'll get into that in just a second. Diffusion is the movement of molecules from areas of high concentration to lower concentration until equilibrium is reached. If you've ever squirted a squirt of perfume or Febreze or something like that into a room, you realize that the odor, the scent, is in one location to start with, but eventually it disperses through the room. So that is diffusion. And we have lots and lots of examples of that out there in the world. This is some physics, it's just the way that molecules work. They move around until they are equally dispersed. So osmosis is a type of diffusion. Osmosis is movement of water across a semi-permeable membrane, like cell membranes, until you have equal numbers of water molecules, unless there's something else at play here. But that is what osmosis is, movement of water across a membrane. So we're going to take a look at that. And then you're going to study about different cell types. So the two main cell types are prokaryotic and eukaryotic. Prokaryotic are also called bacterial cells. So bacterial cells are very, very different from our cells. They are small, they are very simple. There are no compartments like a nucleus in a bacterium, because that bacterial cell is not big enough to hold a nucleus. There are no mitochondria, chloroplasts, anything like that. All the molecules inside a bacterial cell are in close contact with each other, including the DNA. So you'll learn about bacterial cells. You'll also learn about eukaryotic cells. Well, then if they're not bacterial, they're eukaryotic, but what does that include? Animals, plants, fungi, protists. Protists include algae and amoeba, oh my goodness, so many others, and parasites that we have like Giardia, etc. They're all different types of protists. So basically, all the other cell types on the planet are eukaryotic besides bacteria. So you're going to learn about those two cell types and the differences between them when you make drawings of a bacterial cell, a eukaryotic cell, and then also a virus. So I did not include viruses here because viruses are acellular. They are not made up of cells, and that tells you right there that they are not living. Viruses are not living. They're just little packets of genetic information covered by a protein coat. Now, sometimes they have some extra parts, like an extra membrane that they pick up on the way out of your cells. And they have extra parts, tails, and all sorts of things. But viruses are just little packets of genetic information that are used to hijack cells. So they are always parasitic. They always infect host cells, take over that cell, and cause the cell machinery to create new viruses. And eventually that cell will die, no matter what. A virally infected cell is going to die. Sometimes it takes a long time for it to die because the virus can just hide out for a long time. like with HIV, but other times it'll be a very quick infection, like with COVID, and cells are destroyed. So viruses always take over host cells. They're like little pirates. Also, they're very much like computer viruses. So the reason that computer viruses were named that was because they act like actual viruses, right? If you get a computer virus, is that a little tiny microscopic computer that sneaks into your house and comes in your house and gets inside of your computer? No, 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 no. What is it? It's just code, isn't it? It's just code. And it takes over the machinery of your computer and sends out 10,000 email messages to all your contacts, or erases stuff on your computer, or gets into your bank account, or whatever. That's what a computer virus is. Works just like viruses do in cells. So you're going to learn about the structures in cells and viruses because you're going to be drawing some based on the lab instructions. And then you will also identify the ones that you have produced. So I'll talk about that in just a second. All right, let's talk about osmosis for just a minute. So remember that I said osmosis is the movement of water across a membrane. So you are going to simulate a cell membrane with what's called dialysis tubing. And the really important thing to remember with dialysis tubing is that only water crosses it. So you're going to be working with some sucrose, that's sugar. Sucrose, the sugar solutions, and only the water is going to go back and forth across the dialysis tubing. So let's take a look at some tubing, or a membrane in this case, in my two beakers that I have here. Okay, on the left, you see on the left side of this, we have a low number of sugar molecules on the left, but that also means we have a high number of water molecules. On the right side in that same beaker, let's see if I can use my my mouse here. Over on this side of the beaker, we have lots and lots of sugar molecules, those red ones, but we have fewer water molecules. Okay, so, and right here in the middle, we have this membrane. So remember that water diffuses over the membrane. So water is going to move from areas of high concentration to lower concentration until equilibrium is produced. All right, here we go. So what happens? Look what happens over here. The water from the left side moves over to the right side until there are equal numbers of water molecules between the two sides here. Okay, so that's what you should see in your dialysis tubing experiment with the sugar solutions or sucrose solutions. You should see this effect. And when you look at how much sugar is in the different solutions, say you've got a 40% sucrose solution somewhere and a 20%. If it's 40% sucrose, that means there's less water. If there's 20% sucrose, there's more water. Okay, so just remember that. And then you can figure out what happens. All right, so that's osmosis. All right, let's talk about all the labs that you have this week. You have chopping up a potato and look at diffusion of potassium iodide. You have the dialysis tubing experiment, and you have to draw cells, two types of cells in a virus, and then use the keys, dichotomous keys, which I'll explain in a second, to identify what you have. And then you also have to do some work, milestone 1, on your research project, which I'll get to in just a second. So let's start going through these items. First of all, it's going to take you time. In both of the experiments with the potatoes and the dialysis tubing, you have some downtime. You have to wait for things to happen. So that would be downtime, where you're not actually doing anything, but you still have to check on it, come back to it and check on it to see what happened. So the potato experiment is the diffusion lab. You're looking to see how much of each type of potato, each tiny little cube to bigger cubes to rectangles, how far in potassium iodide is diffusing into that little chunk of potato. So how does this work? So this iodide solution changes to a black color. It's normally sort of a light brown color, but it changes to a black color in the presence of starch. Sorry about that. In the presence of starch. Now, you know that potatoes contain starch, right? Everybody knows that. So, how far in does this diffusion happen? You can tell because that little potato cube starts turning black from the outside in. All right? So, that's what you're going to be taking a look at. So that's going to take some time. And really, it took me the most time to chop up all those little potato squares and rectangles in the right sizes. And if you don't get them exactly right, that's okay, because you can fix it in your table. Okay, so don't obsess about all those tiny, especially the tiny little measurements, like half a centimeter. Oh my gosh, they're so little, the tiniest little cube. So anyway, do the best you can. You can always adjust in the table. The dialysis tubing is the osmosis lab. And so this is where you're going to be working with the sugar concentrations in the beaker and in the dialysis tubing, and then you're going to see what happens. Does the dialysis tubing swell up? Does it get smaller? You're going to figure this out. You're going to see what to do when you get started with this. Let me give you one hint about the dialysis tubing. It's going to tell you that you're going to have to pour a solution into the tubing. All right, it's plastic. You have to put it down in water first, and then take your fingers like this on the tubing, and then you can open it. Do not try to open it while it is dry, because you cannot get it open. It has to be wet, and then you just do like that, and it'll get open. It'll come open. All right, so that's just a little tip. For all of these, you need a potato, paper, calculator, and a timer. You know, you can use your phone. Everybody's got a phone now. Wear your safety gear. That potassium iodide is not friendly. So make sure you wear your safety gear, including your eye protection and your gloves. There's about 1+ hours of downtime per experiment. You can get both of them set up if you want to, which I would recommend. But realize that these are going to take some time to do. All right, on page 24 of your instructions, the last column, you should think of it as saying outside the tubing compared to inside the tubing, the solution in the tubing, outside compared to inside. And there are some terms that you have to use: hypertonic, hypotonic, isotonic. So isotonic means that there are the same number of water molecules inside and outside something. Okay, you're comparing two solutions. So let's see, like salt water in the ocean is isotonic to the solutions inside our cells, which is very interesting. We have about 0.09% salt in each of those locations. So that's isotonic. Hypertonic means that there are more solute molecules in one spot than the other. And this is the way that you can think of this. Hyper, more sugar. So if a solution is hypertonic, it's going to have, think of it that way, more sugar in it, and that means less water than the other solution. Hypotonic is just the opposite. There's going to be more. So hypotonic means that you're going to have more water there than you have solute molecules. An example of this, let me give you an example of this. I had a senior microbiology student working with me when I was doing my research, and she was supposed to be counting white blood cells for me. So we'd gotten a blood sample from someone, and we'd isolated the white blood cells, and she had to count them because we had to have a certain number to do our experiments with. So she was sitting right behind me at a microscope, and she goes, "Dr. Sigmon." I said, "Yeah, what, Heather?" She said, "I only had 4 white blood cells in this one little grid that counted, and I went over to the other grid and there were none, and then I went back to the first one and there were none there anymore. What in the world is going on?" I said, "Heather, did you put the white blood cells in water?" She said, "Yeah." I said, Heather, osmosis, remember? She thought about that and she went, oh no. Okay, there was more water outside the cell than inside because it was pure water. So what happened? The water molecules moved inside those white blood cells and they grew and they grew and they grew and they grew until they blew apart. So, yeah, that's how hypotonic works. So anyway, let's see what else you got. Draw a couple of cells. I already mentioned that. And then you're going to identify what you have drawn by using something called a dichotomous key. So a dichotomous key is a way for you to identify something. You can identify birds this way and plants and all types of cells. So what does dichotomous mean? It gives you two choices. So, for example, with viruses, is this a DNA virus or an RNA virus? Two choices. You don't have a third. It's got to be one or the other. Which one is it? Is it a DNA virus or an RNA virus? So then if you choose DNA, is it a single-stranded piece of DNA or is it a double-stranded piece of DNA in this virus? And you choose which one it is. So that's a dichotomous key. Two choices and you follow the path until you get your identification. It's very easy. You'll get the hang of it. A reminder about next week. Next week, your experiment has to be started by Thursday in order to finish it because you've got to germinate some peas and it takes a while. You have to soak them for a while and get them all prepped before you can actually do the experiment. So I want to warn you about that ahead of time. And you're going to need plenty of non-chlorinated water. You cannot use tap water to do that experiment. Now, all of us have bottled water now, and none of that bottled water has chlorine in it. So you can use bottled water. You can use filtered water, but not water straight out of the tap. You will kill those peas, and then you won't get any results whatsoever. So just saying. Another thing to remind you of is to use the lab report forms. This is extremely important. I just finished grading the week 1 labs, those 2 labs that you did. And quite a few of you did not use the lab report form, so you didn't answer the questions. You didn't produce a graph in the second one. The second Excel graph that you needed to produce. You didn't answer the questions. You didn't synthesize the material. You just did the experiments and gave those to me. And that's not what you need to do. You need to do the actual lab report. So maybe I'll have to send out the instructions again. I think I'll do that so that you can find those lab report forms. They're kind of hard to find, which is why I've already sent out one email about it, but I'll send out another one. Include everything in the report. I shouldn't be looking at 5 files. I should be looking at 1 file, 1 lab report for each thing that you have to do. So for this lab, there is literally 1 lab report for you to do. Include everything here, including the photos and Excel graphs and whatever you have to put in, your answers to your questions and all that. You'll have tables this coming week. Okay, so make sure you put everything into that 1 lab report. Okay, I'm going to go over a couple of extra little things to help you. Only if I get to the next slide. Okay, for the rest of the term, you're going to be filling out tables like this. And I want you to notice something. Okay, this is one that you have to fill out this week about the potatoes. You know, measuring 2.5, 2.5, and 2.5. Yeah, that's a cube. So anyway, you have to do all these calculations, right? And that's columns 3, 4, 5, 6, 7, 8. Those are calculations that you are doing. That's not what's so important. I will be looking to make sure that you do those right. Okay? But that's not what's important. What's important in any of these tables from now on is that last column. You see the red arrow? That's what you're trying to get to right there. that information. All those calculations all the way across in a row, that will get you to that last column, and that's what is important. That's what you're supposed to be looking at and figuring out what is going on. And in this case, you're figuring out how diffusion occurs in a bigger slice of potato versus a smaller slice of potato. The whole point of this is for you to determine that cells can never be very big, because diffusion has to happen at a certain rate for the inside of the cells to get the right nutrients and get the right amount of nutrients and to get rid of the right waste. And the bigger and the bigger and bigger and bigger that a cell gets, then the less diffusion is actually going to take place compared to the volume. So that's why you need that last column. And that is true for every table moving forward. And really, every table anyway, whatever you're talking about in science, you're trying to get to that last column because that's your results that you're looking at. Okay? Just wanted to point that out. Wanted to talk about COVID for just a minute, since it is a virus. And we have all been inundated with information about it for the last 2 years. I just wanted to give you some specific information about it. The coronavirus is a specific type of coronavirus called SARS-CoV-2. That's the name of it. That's the actual name of it. So what the heck does all that mean? SARS means severe, acute, respiratory syndrome. Severe is a bad one. It's a bad disease. It's going to make you really sick. Acute happens right away. Get infected, you get sick. Respiratory, yep, in your lungs. Syndrome, collection of symptoms. That's what that means. Okay, and then COVID, corona, virus. You got it? 2, second one that we've identified. And then what the heck does COVID mean? COVID-19, you know, it was discovered in 2019, so you get the 19. COVID stands for corona, C-O, virus, V-I, disease, D. Okay, so that's the disease, but the virus's name is SARS-CoV-2. The virus is an enveloped virus, which is very good news for us. It has a piece of our membrane as it makes its way out of our cells, and that membrane is a lipid, phospholipid membrane, which means that it is easily dissolved with alcohol, and so that's why hand sanitizers work so very well against this virus, because of that envelope. It's an RNA virus, and the vaccine is actually based on the RNA. So we take a little part of that viral RNA that codes for one of the proteins. We put that in our muscle cells in the vaccine. Our cells start making that. Our immune system reacts, and then we have protection. The spikes that you can see on the outside of this virus are used for attachment to the host cells to help them get into our respiratory tract cells. For more information, go to the CDC website or ask me questions. I love to talk about it. So if you want to ask me questions, go for it. All right, and then the final project, milestone 1, is due this 3rd week. Go to the Ask a Biologist website. You should have already chosen one of the 4 experiments, or I gave you some ideas of some other ones. I forgot. One's the egg one. Another one is one that has to do with ice and density. You could do something else. One of my students did a composting one. There are other ideas out there. But go to the Ask a Biologist website, choose one of those experiments, figure out a research question. So you're going to have to ask a good question that you want to examine. And you're going to have to find some background information on this. So why the heck is this important in the grand scheme of things? Why do you need to know if saltwater ice has little cracks in it when it forms? Why is it important to know that you have more pain receptors in certain areas of your body than other parts of your body? You know, things like, why is this important? And it is important. You can't just say because it's on the Ask a Biologist website. You have to figure out something. And what is known already about this phenomenon? Oh man, when you do actual research, that is probably what takes you the longest of anything, because you have to read everything in the world that you can get your hands on about your topic. But you don't. But anyway, find out what you can about this phenomenon. The format is important, 1 to 2 written pages. This is not 2 sentences, your research question, and then the background information about why this is significant. Nope, 1 to 2 pages of written information. And of course, cite your sources like you're supposed to, okay? Because you're looking up background information. You didn't know that beforehand, so make sure you cite your sources. And as always, if you need anything, please let me know. This may be a confusing week for you. If it is, I'm here for you. I will answer your questions. There's my email address down there, m.sigmon@snhu.edu. Just let me know. Okay? I hope you have a wonderful week and get started as quickly as you can.